Hello friends, in this video we will explore cellranger. We'll do the practical hands-on session. Okay, so cellranger is a tool developed by 10x genomics to convert the raw sequencing data to ultimately the count data. Okay, so in the last video we talked more about the cellranger theory, like how we explored how cellranger works, and we saw different subcommands of cellranger and different flavor of cellranger workflow. So now in this video, we will mainly do the hands-on. So first step first, let's download the tool. Okay, so we can go to so the website here. Okay, so for example, you go to the cellranger website, and in the download section we'll get in this undergo in the downloads we get the download links. So cellranger we do not need to install, simply we need to download it. So we can use this curl command. Okay, so to download it, so let me show you. I'll not download because already I have downloaded it. Copy it. So let's say in this example I want to download it. Okay, so you can simply type this curl command. Okay, so now it will see, it will start downloading. Okay, so once you see, it will download the cellranger 710 tar.gz file. So this version I'm using. Okay, so we can now extract it using this command tar xzpf, then this one cellranger 7.0 tar.gz. So this will extract it. So I'm just commenting it, I'm not executing it because already I have done the extraction here. So you see the cellranger is now extracted. So if I go to the cellranger here, okay, this is a tool. So there is a bin folder in there. Okay, so where the executives are there. So now you see the cellranger execution are there. So directly you can use dot star means in this current directory we can run the cellranger tool. So now you see that we have different sub commands are there, like count, multi, like that. So we will be using count. So what we can do, we can now give count, and when we type simply enter, so you see what it is expecting us. Okay, like we need to provide a transcriptome, the reference genome, and then we also need to provide the FASTQ file, but here the options are not clearly shown, so we can say my hyphen h. So this will give you all the help. So we see what we need, like we need to provide the FASTQ file, the project, we need to give some sample, which lanes, lot many information are there. But yes, so this is the way the tool works. We'll talk about some parameters here. So now, so if I want to use this tool, so I just have to use this path, right? What is my current working directory, like home, intel, this thing being within that cellranger, this is the path we will be using. Okay, so now let's go. So we have downloaded our tool from cellranger. So now we will be downloading the data. Okay, so I'm just following the same example that is given by the cellranger tutorial. So we are looking into one of the publicly available data set on let me zoom it a little bit. Okay, on the 10x genomic support side, and basically this example includes 1000 pvmc data set from human peripheral blood mononuclear cells, and it consists of different subtypes, the lymphocytes, particularly T cell, B cell, NK cells, as well as monocytes. Okay, so we can get the data from this by wget command. Okay, so already I have downloaded it, but I'll just show you. So if you see wget like this, so it will now connect it, and it will take some time to download your data. I'm just canceling it. So so what does the data look like? Okay, so now you see the data will look like this. Okay, so basically it consists of 6 files, and you see the I1. So these are the index file, we do not need them, but we need so it's a paid in data R1 R2 R1 R2, and one ran on len 1 and one ran on len 2. So same sample, but it ran on multiple lens L001 and L002. Okay, so apart from this data, since we need to align this FASTQ file against a reference genome, we need to download the reference genome. So basically, cellranger already provides pre-built reference genome, and we will be downloading that. Okay, and yeah, so the reference genome data actually looks like there will be a FASTA file. This is the genome sequence, and it is already indexed. And we also need an annotation file which will tell how where the gene is present from start to end. So we need that gene location information, so which is there in the GTF file, and the pickle format of the same information. And we also need the index from star to, because I told cell ranger uses internally star to align. It is a reference ever aligner for aligning the FASTQ file against this reference genome. So star building an index already takes some time, so that's where it already pre-computes for us, and it provides this compressed the genome reference file. So if we can directly go to their website and download the, for example in this case GR38, okay, and if we want some custom reference, then there is a way we can build this kind of format. But for now, just we will be using, we can download the reference data pre-built from their website. So if we go over here in the same example section, so we will see in a pre-built human references. So we can use the wget command to download it, and then we can extract it using the tar command. Okay, so I have already downloaded it, so I'm not showing again. So if I go to my download folder, you see I'll have this pbmc, the tar file, and reference data. And using that, I have already extracted, and I have extracted in this folder, data folder. So we have now see the two folder. So let me show you the folder structure. So now you see that this pvmc, in this folder we have this many FASTQ files, and this is my reference data. And within that we have the genome FASTA file, the genes GTF file, and the same in pickle format, and the JSON file, something like that. And most importantly, the star index file. So this is what the reference file. So now if I look my present working directory, this is where my actually the FASTQ file and the reference file is occupied. Okay, so now good. So we have the FASTQ file, we have the reference, and we have already downloaded the cellranger. We are good to go. So let's first run the command. So okay, cool. So let's run here. Okay, I will be running, I will not mixing that data and download. So let's run here. So now I have to use the cellranger, right? So the cellranger is present where, in this current directory we have the cellranger, then we have the bean, then you have the cellranger. Okay, so now first is now count command. So count command, so it is showing that it is the mandatory argument is required. The first argument is id. Okay, so this id is nothing but the output folder. So let's say we call run one. Sorry, so let's say this is my output folder id. So now what we need transcriptome. And transcriptome is what? Basically the reference data. So which is present where? In this current directory, it is, we have inside the data, we have this reference. This is my reference data. Okay, so we also need what? Let me comment it out. Okay, so cellranger bean, cellranger count. Okay, just to show you the help file. So we need the FASTQ file will be provided using, okay, FASTQs. Okay, not FASTQ, FASTQs. Okay, so here we provide the input to the FASTQ file, and then we provided the transcriptome, and we provided the output folder unique ID. So now apart from that, we need to provide the sample, which is a prefix of file names that first used to select. Okay, and some other informations are there. Okay, so let's do this up to this one. Okay, so we have now we provided the transcriptome, so now provide the first cues, so which is also there in the data, and pbmc, in this place we have the first queue file. So now we need to give sample, I guess, right? So samples or sample, we need to provide a sample by which cellranger will identify the first queue file. If you remember the this one, right? uh, the first queue files are here, right? So this is the right. So let's use this name, okay, as the sample name, pbmc 1k v3, pbmc 1k v3. Okay, so this will be our, okay, the sample name. So when we do it, right, it will run it. Okay, so it will take some time to run it. Okay, maybe more than, for in my laptop it took a lot of time. Okay, so I'm just canceling it because already I have run it, and I will show you the data. But we can also use this parameter local mem where we can set the local memory. Okay, and we can use simply 675, okay, like that, or 10 GB or on 100 GB. So you just provide the number, not, you don't need to write G because it's already in the set of GB, right? So you just have to give it how much memory we want and how many cores we can use. Okay, and also maximum jobs, how many memory per core. So those, basically the memory and CPU configuration we can set. So higher is the memory and higher is the CPU, it will run very very fast. And most importantly, the include intron parameter can be set to true or false, whether in the downstream analysis we will be including the introns or not. Yeah, and some other informations are also available, like we want to select the first group from lane number one, like that. But in this example we do not need that. We just provided the transcriptome, basically the reference transcript genome, and we provided the first few files, and and we provided a prefix which by which our, it can identify which samples to pick up. Okay, so cool. So I will show you the output file. Okay, so the output file will mainly look like this. Okay, there will be many folders and some files will be there, but most important file are the QC metrics in this HTML file. So let's just click it. Okay, apart from that, the final data will be there in this filtered feature barcode matrix. I told you that we'll have three files. One is the barcode TSB file, one is feature file, and the matrix file. We'll talk about the format later on. Okay, but yeah, so this is the final data. But let's look at the QC metrics. So now this is the HTML report will look like. First, if some alert messages are there, it will provide. So in this case it showed we have used the intron mode. Basically I did not specify include intron false, so that means the introns are included in the analysis. Okay, so it will give you two tabs in this example, but there will be multiple tabs depending on what kind of algorithm, what kind of data you had, like antibody, CRISPR, like that. But now this is the gene expression data, so we have two tabs. First is the summary tab, and then we have the gene expression tab. So we'll talk about first summary. So this will give an overall idea about how many estimated number of cells are there in this example data. So there are 1231 cells are expected, and on an average, how many reads were there. So mean reads per cell, on an average 54,000 reads were detected inside a cell, and but the median value of number of genes detected is 3,000. See, you see that the mean and medians are very different, so it is a skewed distribution. Okay, and here we will get lot of sequencing parameter, and if you hover on the question mark, it will describe each of this parameter. So, and basically these are straightforward, how many reads were there and how many were short reads, so that's why it was not included. So in this case there was no short reads, so all were included. And now you see the first step what cellranger does is it checks for barcode validity, right? And in this example 97.4% were valid, that means three remaining around 2 to 3% were discarded. Similarly, how many UMIs were valid? In this case, 99.9 UMIs were valid. And then we also talk about sequencing saturation. I'll talk about that. That is around 70% here. And next important one is the these three parameter, like Q30 bases in the barcode. So means how many bases are there, how much percentage of bases of barcode follow good quality Q30. That is 94%. That means remaining 6% of barcode are having less than Q30 quality. Same, what is the Q30 in UMI? That means 92.7% of bases of UMI are above Q30 quality, and 90% are actually above Q30 in the reads. So this way you get a lot of QC parameters, but most important I look for is this one, and I think this one only, and this one. So this will give you how many barcodes are retained, like that. Okay, so the next step is like mapping. The mapping QC, it shows how many reads were mapped to the genome of the total reads, and of which how many are confidently mapped, and then how much confidently mapped to the intergenic region. So basically these 4% will be discarded, and how many mapped to intronic region, and how many to exonic region, and total overall transcriptome how much it is aligned. and how what is the percentage of reads mapped to antisense gene. So these will also be discarded. So this way you, so in your actual good QC data, ideally there should be less amount of this one, so that more higher amount of higher should be the mapping rate, we can say. Okay, so once we have the sequencing and mapping information, now we get the cell QC. So we got the sequencing QC, we got the mapping QC. Now our cell is called, we get that QC. So here you get a barcode rank plot. So basically, this is a cumulative frequency kind of plot where the y-axis contains the number of barcodes, and while the the x-axis contains the number of barcodes, and y-axis contains the number of UMIs. So now let's take this first point. What does it mean? It corresponds to the UMI total. How many UMIs are there? Like 100k UMI. Such a high number of UMIs are there. So now we check how many cells that much UMI is detected. So in this case, only there is, let's say, one cell is there. So where this much amount of barcodes are there. So now as you move rightward towards right, you can see that less and less number will have this much amount of barcode. So let's say example 10k parameter if you choose, so you go like this and you put a drop down here. So that means only let's say hardly 300 or 400 barcodes or cells are there which are having more than 10k UMS. So that's where this graph will always go down. But as I told in the last session that not all the drops can be called as cell. Okay, so in order a drop to be called cell, it has to follow a proper gene expression profile. Okay, so that's the logic cell ranger uses to differentiate from the actual cell from the background. So that's where you will see the difference, the percentage. So for example, in this parameter, whatever the drops were there, all of them were 100% cell. Okay, so when you, and here also in up to this blue line, all were 100% cell. But if you come here, you see there is a drop, right? So let's say this point shows that of the total 22 drops, 21 are called cells. Remaining one, that is not called as cell, but that is called as barcode. Now if you come down here again, let's say 68%. So here, see, out of the 22 cells which are having this much amount of UMIs, 15 is actually cell, where the remaining is not cell, that is background. But and after that, you see all of them are actually called as background only. So that's where you see a difference between cell and the background. And ideally the this the graph should be, there is a steep decrease should be there, it's steep fall should be there, so that we can clearly segregate the cells from the barcode. It should not look like it is going smoothly and it is decreasing. So that means both cell and barcodes are slowly decreasing. So that means there is no clear cut boundaries there but cell and barcode. So such is not a good quality data. So ideally in a good quality data, there should be a strict difference between cells and barcode, which we can get it from this barcode infer plot. There should be a steep, down in this case. Okay. So this is summarized here, like how many estimated number of cells and fraction of reads in the cell, and what is the mean reads per cell. So this is the same number what you see here. What is the median here, and how many on an average how many UMI were detected per cell. Okay, and so how many, see, this is very important. So reads, number of reads is different from the number of UMIs. Okay, so here the reads are 54,000 in that range, but the UMIs are in this range. So again, we can say the median read where so I think I did a mistake here. So this is the mean reads per cell, but this is actually median UMI counts per cell. So we cannot directly compare this number. So if you would have some, let's say median would be in somewhere, let's say in this range only, let's say 30,000 or 40,000 range, let's call. So then we can compare that, the UMIs and the reads would be different. And similarly, median genes per cell are in this range 33 to 35 are there. Okay, and total genes detected is 24,000 genes are detected. Okay, and then you'll get a QC on sample. So the sample name we used, the output name, sample ID, and what chemistry it used, and whether we have used this parameter include intron or not, and what reference version we use. So those many basic information it provides. Okay, so we got the sequence mapping and cell QC and finally the sample information. So then we go to the next information where the this is the important information where we will see how the cells are clustered. Okay, so now since it's a multi-dimensional data, right, we have so many genes are there and so many cells are there, and we cannot draw a multi-dimensional plot, so we need a projection plots. So there are different way we can do a two-dimensional projection, like principal component analysis, PCA, TSNE, and UMAP. These are the three popular method that we use, but in this case the TSNE is shown. Maybe in the next video or somewhere I'll explain the difference between TSNE and UMAP. But to give you a brief idea, TSNE will, it's a statistic stochastic based method and tries to cluster the cells into different, clusters. Now each dot here is one cell. Okay, and the TSNE is very good for visualization purpose. The disadvantage is like is not, it doesn't retain the global structure. So what I mean global structure means, within a cluster, the cell to cell, the distance is meaningful. So that means in this, let's consider this cluster, this cell and this cell are farther away from each other. That means the expression profile of this cell and this cell is different because they are farther within the cluster, and all these cells are having same kind of profile. But TSNE does not retain the information between the cluster. So that means we the distance is meaningless in TSNE plot of the two clusters. Okay, so that means we cannot say that this cluster is farther from this cluster means they are different. Okay, it's not like that because TSNE doesn't retain the global structure. Okay, so within cluster distance is meaningful, between cluster distance is meaningless. Okay, so that's where we have alternate better method UMAP, which does the same thing but where the global information is retained. So where within and between distance on the UMAP plot is important. But just forget about that. But in this example, it shows the TSNE cluster plot, and here the same clusters. So we are showing that in this case, TSNE detected 7 clusters here, and here we are same thing, but we are color coding with respect to how many UMIs are detected. You see the red dots means they have higher number of UMIs, whereas the blue dot are, blue cells are having very less number of UMIs were present in those cells. Okay, so now we got the cells. So in the next, what it does, like it also compares every cluster from the other cluster and do a differential gene expression, and it gives which are the genes that is differentiating this cluster from rest all of them. Okay, so that way we get the top genes. So in this case these are the genes which are specific to cluster 1, so we can say that these are cluster 1 specific biomarkers. Okay, so if you double click here, so you will get those genes which are specific to, cell cluster 2. Similarly you get cluster 3 specific marker, cluster 4 specific marker, like that. So every cluster will have its own signature markers. So now if you know about this gene, like let's say this AK5 genes and APBA2, let's say this is T cell, so you can use this information to then annotate like, oh, okay, so this is the marker that is differentiating this cluster. So and these markers are specific to, let's say, T cell, so probably I will call or I will annotate this cell into the T cell. These all are done by the downstream packages like, Surat and all. But to give you a rough idea, like this is what the single cell data looks like, and this is what we do. Okay, and last we'll also get the sequencing saturation information. So this tells, uh, I'll just read it for you. Sequencing saturation is a measure of object library complexity, and it approaches 1.0. So how complex your library is, it should be near to that. So in this case, in our example, we have got 70% somewhere around, the sequencing saturation. And also it will give you median genes per cell. This is a cumulative plot kind of thing. If you read about that, the slope of the curve near the endpoint can be interpreted as the upper bound to the benefit to the, means like as we increase the sequencing depth, how the number of genes increases. This is kind of some other QC plot we get. Okay, so this is one output file. Okay, so then I told like what we'll get. Most important thing is this file, right? the filtered feature barcode matrix file. So basically this file will contain three files: the barcode file, the feature file, the matrix file. So what does the format look like? Okay, so I'll show you the format. Okay, so the barcode file will contain, you see, this is the so the barcode file, barcode.tsv file will contain the barcodes. Okay, so these are the 12 base pair barcodes usually, and there will be, suffix -1, -2, -3, depending on what same barcode used in multiple gem wells, right? So in that case it differentiates like -1, -2, like that. So these are the list of barcodes. And the second file, feature file, is a multi-column file which contains the information about each of these genes. Now we have the matrix file, and since this is a very huge data, and it is stored in a very unique format, this format is called as mtx format. Okay, mex format. It is stored in matrix, some, I don't remember the full form right now, but it is the mex format. Basically what it does is, like, it on that, the first three line are the metadata. Okay, and it tells how many genes are there, and how many cells are there, and how many UMIs are there. So how many features are there, or how many barcodes are there, and how many UMIs are there. So now in the first example, let's say 32709. So what does it mean? This tells that 30709th line from this, the feature file. So in this case, the line number 32709, or the index number 32709, is this gene, MIRI gene. So that means this gene, whereas one here indicates the which line from the cell barcode file. So that means this barcode. So what, and the 4 is the number of UMI supporting. So for the first line, what we can infer, that means the MIR1302, this gene, in this particular cell, there are 4 UMIs are supporting. Okay, so like that. So this is a very compact way we store the information into these three files, and this, we need all these three files, and we feed it next to the pseudo package. Okay, so now there is also a folder called raw feature barcode matrix you will get, but this contains both cell and background. But filtered will contain only the cell information, and that's where we are using this filtered feature barcode file. And also we'll get some analysis file, whatever we see in this QC matrix we saw, right? PCA, TSNE, all these analysis file we get. Apart from that, instead of this mtx file, the same information is stored in a binary format to space, reduce the space. And the binary format is htf5 format or h5 format. We cannot see it because it's binary format, but this can also be used to in the downstream process of, like, let's say Surat or any scanpy we can use this binary file as well. Okay, and also there is a, it gives you the clue file. So 10x genomics has one more data visualization tool. They have developed cloud browser, so we can you load this file into that browser and see it. So it's not part of this video. I'll just show you, just like, the cloud file. So you need to download this cloud tool from the 10x genomics. So once you download it and you load the data, but only, like, it does a lot of things we can do. Same thing, it does the clustering, and there are 7 clusters detected, and which are the genes that are differentiating. But the interesting, like, we can add those genes into here in this plot as well. The color code, the features, a lot of things can be done. Maybe some other video I'll talk about the loop browser here. But yeah, so this file, so sorry, not cloud, actually loop browser, my bad. But yeah, so yes, so these are the data you get, and also you get the bam file and index file. Also in this file I have just done a dummy one. Okay. Yeah, I think that's all I had. So if I summarize what we did, like, we saw, we downloaded, we extracted, we run this simple example. So we gave these many parameters: cell ranger count, id, transcriptome, first q, sample. And we can explore other parameter, like include intron, local course, local memory. Then we saw the output, and mainly we explored the mtx format. Yeah, that's all about the cell ranger. So in the next video we will talk about Surat. Okay, so we'll be splitting that video into multiple sub videos, but we'll start with how to load this excuse me, sorry, how to load this data into Surot, and how we'll explore the Surot object in the next video. I hope this video was useful, and if yes, so please share among your network. And if you are first time and you have not yet subscribed to my channel, so I request to subscribe it. And yeah, thank you. See you in next video.